One potential mechanism that may operate to produce stasis within species is the fact that there are limits to variation. After all, variation is what selection operates on, so without variation we could not have evolution. And indeed, there can be limits to the production of variation, at least variation that yields viable genotype and phenotype combinations. As part of this discussion, it's worth considering how genetic variation emerges. Recombination of genes and new mutations create variation. However, both of these may lead to deleterious effects as often as they create beneficial changes. As a result, many new genetic forms within a species may be quickly eliminated from the population. For instance, recombination may disrupt necessary gene combinations or bring together genes that have a negative effect. In addition, new mutations are also often detrimental to the organism, as they occur randomly, and most random changes to functional genes interrupt proper function. This is especially true if larger changes to the genome occur, such as chromosome deletion, chromosome duplication, and chromosome inversion, and these changes are often fatal. What this can mean is that especially if a species exists in an unchanging environment, the standing genetic diversity may be largely exhausted for that species. This is because the species may be as well adapted to the environment as it can be, without sacrificing additional fitness to allocational trade-offs. Considering all of this, it is rare for new beneficial genes or gene combinations to occur. However, the circumstances where a species lives in an unchanging environment are expected to be rare, as fluctuations in the environment are common over short and long timescales. But this is one scenario by which variation remains limited, leading to little or no net change in species through time. At present, this mechanism is not thought to be one of the chief mechanisms of stasis, partly because tremendous amounts of morphological variation can be introduced within species over short periods of time, at least in situations manipulated by humans. These include experiments on butterflies in the lab and the rampant changes that have occurred associated with the domestication of dogs. However, the mechanism certainly plays some role in mediating stasis, and hopefully data collected in the future will help better quantify that role.